system. Then lymph we're going to discuss, and also the last one will be function of platelets. So one by one, we're going to understand, and also we're going to learn that how the question should be written in a very simplified form, and also what are the major and most important headlines that should be covered under these topics. So first we have the major function of blood. So this question is a very important one, and also it can comes in various type, like the examiner can ask you that state the major function of blood, and they can ask you that define blood and describe the major function of blood or properties of blood. So in those way they can ask you about the function of blood. So we're going to understand that what are those basic points in which the student should answer these topics, right? So first we have the breathing function of blood. So as we know that blood carries oxygen and also CO2, right? So blood has a very important pigment that is known as hemoglobin, and with the help of hemoglobin, the blood carries CO2 and oxygen. So that's how it performs the breathing function of blood, right? Now, next we have the tropic function of blood. So as we know that blood delivered CO2 from all the tissues. As we know that all the tissues perform various metabolic activities, and through those metabolic activities, they are able to produce this unwanted product, that is CO2. So hemoglobin pick up those pick up CO2 from the tissues and delivered to lungs, right? And from there, hemoglobin pick up oxygen and delivered oxygen from lungs to tissues, right? So this is how the function of the second function of blood is served, that it is a tropic function of blood in which the CO2 pick up by the hemoglobin from tissues to lungs, and oxygen from lungs to tissues, right? Okay, then next we have the excretory function. So in excretory function, we learn about that waste product from in the tissues that are produced during various metabolic activities are removed by blood and also carried to excretory organ, and those excretory organ involved like kidney, skin, liver for excretion, right? Then next we have the hormonal function. So as we know that blood used to carry various hormones and various enzymes. So the blood functions to transport it is the basically it is a transport medium, right? Then next we have the temperature regulation. So because of its high specific heat, and the blood is responsible for maintaining thermoregulatory mechanism in the body. And what is thermoregulatory mechanism? It is the balance between heat loss and heat gain in the body. So by heat loss and heat gain, the blood is able to perform the function of temperature regulation also. Next, we have the function that it help in the maintaining the acid-base balance of tissues. So we know very important content, that is plasma protein, right? So plasma protein and also hemoglobin, because hemoglobin also has a protein, that is globin. So plasma protein and hemoglobin both act as a buffer and help in the regulation of acid-base balance, because both plasma protein as well as hemoglobin both are amphoteric in nature. Amphoteric in nature means depending upon the circumstances, they can act as base or they can act as acid, right? Now, next we have that it is the supporting one for the water electrolyte balance also. So as we know that blood is help in the regulation of water content of the body. As we know that blood carry plasma. Plasma is 55%, right? And the formed elements are rest 45%. And the major product of plasma is water, right? Now, next we have hemostasis function. So as we know, hemostasis means the maintenance of internal body environment, right? So as we know that blood is a fluid, so that's how it also help in maintaining the internal environment of the body. Now, last we have the function that it is the one who protect the body from bacteria and other organism that can cause disease or other abnormal condition. So as we know, blood play an important role in the defense mechanism also. How? With the help of the cells, and those cells are WBCs, right? So WBCs are responsible for this function, and specifically neutrophil and monocyte, which engulf the bacteria with the process that is known as phagocytosis, right? So lymphocytes are also involved in the development of immunity, and eosinophils are responsible for detoxification, deintegration, and also for the removal of foreign protein, right? So majorly the WBCs are those cells which are responsible for providing defensive function to the body. So these are the important function of the body, and this question can come from 5 marks to 10 marks. Okay, and if it will come in 5 marks, so you need to state 5 to 6 function, and if will come in 10 marks, so you need to state all the 10 major function. And one more thing, that these are the necessary headings that you should write in the answer, and along with these headings, like you also have to explain them. For example, if we are talking about the breathing function of body of blood, so you have to answer that how it is performing the breathing function. So you will write that hemoglobin is a pigment which used to carry oxygen and CO2, right? So you have to explain all the function under these headings, right? So we're going to have a recap. So in recap, what was the first function that we have discussed? That was breathing function. Then next function we have discussed about tropic function, right? Then next function we discuss about excretory function. So breathing function was a function in which the hemoglobin carries CO2 and also oxygen, and in trophic function, it is the medium for providing a transportation from lungs to tissues and from tissues to lungs. In excretory function, we learned that waste product formed in the tissues during various metabolic activities are removed by blood and carries to excretory organs like kidney, liver, and spleen. Then fourth function, we learned transport of hormones and enzymes. So hormones which are secreted by ductless endocrine glands, right? For example, the adrenal gland is there, pituitary gland is there, right? So hormones are released directly into the blood, and the blood transport the hormones to their target organ tissues, and also blood also used to transport enzymes. Then fifth function we have regulation of water content, or we can write regulation of water balance. Then sixth, we have regulation of acid-base balance. That also we have learned the role of hemoglobin and the role of plasma proteins. Then next function we have regulation of temperature by the mechanism of heat loss and heat gain. Then storage function is there. So what is storage function? Water and some important substances like protein, like glucose, like sodium, right? So are constantly required by body. So blood serves as a ready-made source for these substances also. And these substances are taken from the blood during condition like starvation or fluid loss or electrolyte loss, right? Then we have defensive function. So these are all the major function that is performed by blood, right? Then hemostasis function also. So this is all about the major function of blood. Now, one question already we have discussed, and this question can come from 5 marks to 10 marks, right? So depending upon the marking, the student have to answer where if the question is coming in 5 marks, so you need to write 5 to 6 points. And if it is coming under 10 marks, so you have to write all the 10 important points. Okay. Now, next we have the erythropoiesis and factor controlling it, and also the role of erythropoietin and RBC lifespan. Okay. So these are the various stages of erythropoiesis, and also we need to write a short note on its regulation, right? So understanding about erythropoiesis. So what is erythropoiesis? Erythropoiesis means, erythro means RBC and poiesis means formation. So erythropoiesis means formation of RBC. Right? So first you have to describe that what is erythropoiesis. So erythropoiesis is the formation of RBCs, right? Now, next we have the various stages, right? So this is the first stage, that is known as PUSC, or it is also known as hemocytoblast. Right? Now, next stage is proerythroblast. Right? Now, next stage we have, this is early normoblast. Then we have this one that is known as intermediate normoblast. Then these stages both are early normoblast, late normoblast. And also in late normoblast, we have early stage, that is the part 1, and also we have late stage, right? Then finally we have reticulocyte, and in the last we get erythrocyte. So these are the various stages that occurs in the process of erythropoiesis, and one by one we're going to learn about the stages, about all these stages, right? So we're going to learn about some important points. The first will be cell size. The second is nucleus. The third is cytoplasm. And the last is division, mitosis. Right? So these are the various stages, right? So if the question over, the erythropoiesis will come. So first you need to answer that what is erythropoiesis. So erythropoiesis is the formation of RBC. Then you need to answer that what are those sites in which RBCs can be formed, right? So as we know that RBCs are formed in bone marrow. So you have to write that RBCs are formed in bone marrow, especially in the flat bones and in the long bones, right? So all these things you need to answer. Then you will answer that what are those various stages in which the erythropoiesis will come, right? So this is the first stage, and the first stage is known as hemocytoblast, or the another name is PUSC. PUSC stands for pluripotent uncommitted stem cell, right? Why this is known as uncommitted stem cells? Because this is the mother cell, and this stage is not committed to become any one particular, right? Because all the cell, whether it is WBC, whether it is platelets, whether it is granulocytes, A granulocytes, whether it is T cells, B cells, NK cells, all the cells are derived from this pluripotent uncommitted stem cell, right? So next stage, that is proerythroblast. Now this stage is also known as committed stem cell. Why it is known as committed stem cell? Because now the cell become committed to form the RBC only. Right? Now, next we have early normoblast, then intermediate normoblast, then finally late normoblast, right? So in late normoblast, we have early stage and late stage, and finally we have reticulocyte and erythrocyte. Now, if you can look for the difference, so we can see that the size of this mother cell is very big, right? And the size of the mature RBC is very small. Okay, so now the cell size decreases as the stages will proceed. So cell size decreases. And talking about the nucleus, So we can see in the mother cell, the nucleus is present, but in the RBC, in the mature RBC, the nucleus is not present. And we can easily see that in which stage there is complete degeneration of nucleus. So it occurs in late normoblast, right? So the nucleus size decreases and the complete degeneration occur in late normoblast. Right? And also we can see the size of the nucleus is decreasing. So the size of the cytoplasm is increasing with each stage, right? So as we can see, the size of the cytoplasm in the mother cell is very less. Now in proretroblast, it is increasing. Then in early blood, it is more. Then intermediate blast, it is all more. Then with each stage, the size of the cytoplasm is increasing. Right? Then talking about the mitosis, about the cell division. So as we know that if the cell is having no nucleus, so there will be no division. So basically the division of the cell in RBC stops at late normoblast, right? So in all these stages, the mitosis will be there, but the mitosis will stop at late normoblast. Right? So this is all about the erythropoiesis. Along with this, you can also write that two factors. The first is vitamin B12 and the second is folic acid. Both are responsible for the maturation of RBC. If anyone, whether vitamin B12 or folic acid, if the body is having deficiency of vitamin B12 and folic acid, so the maturation of RBC will not occur properly, right? So all these things are very, very important, and all the students need to cover about this, okay? Then second, we have add a short note on its regulation. So now we're going to discuss about the various things about the regulation. So talking about the regulation part, so as we know, in regulation, we know about general factors, and that is basically hypoxia. Then we have spatial maturation factor, right? So in spatial maturation factor, we have dietary factors. Then we have Cassell's intrinsic factor, right? And the last we have extrinsic factor. So one by one, we're going to cover all of these, right? So in general factor, we learn about hypoxia. Now, what is hypoxia? So whenever any person goes to high altitude, right? So what happened there? There is low partial pressure of oxygen, right? So if low partial pressure of oxygen is there, so what will happen? Low PO2 will be there. So what will happen? The condition of hypoxia will develop, right? So as the condition of hypoxia will develop, so there will be low perfusion of O2 to tissues, right? So if there will be low perfusion of O2 to tissues will occur, so what will happen? Two organs, mainly liver and kidney, they will start producing, if the condition of hypoxia will arise, they will start producing an important hormone, and the name of that hormone is erythropoietin. Now erythropoietin will stimulate bone marrow to produce more number of RBC. So if the count of the RBC will increase, then again this condition of hypoxia will get recovered. Okay, so this is all about the regulation, about the general factor, right? Then talking about spatial maturation factor. So in spatial maturation factor, we learn about the role of dietary factors, right? So in dietary factors, we have proteins, we have iron, then we have manganese, then calcium, and at the last we have vitamin C and vitamin B12. Right? So protein help in the globin formation, right? And globin is very, very important for the hemoglobin, right? Then iron, manganese, cobalt, nickel, they help in the heme formation. So they are helpful for the heme formation and protein for globin formation. Right? Then calcium. Calcium is responsible for increasing iron absorption from GI. Then last we have vitamin C and vitamin B. So they help in synthesis of nucleic acid. Right. Now, if the question over the erythropoiesis will come, then all the student will need to tell that what is erythropoiesis, then what are those stages in which erythropoiesis, in which RBC will develop, then you need to explain about cell size, nucleus, about cytoplasm, about mitosis, and also one thing that hemoglobin appear as intermediate normoblast. This is the most important thing that you need to state. Hemoglobin appear at intermediate normoblast. Okay. Then in regulation, you need to draw this chart. This one is important, in which you will explain hypoxia and also spatial maturation factor, in which we have seen dietary factors, then Castle factor, intrinsic factor, and extrinsic factor. So Castle factor is basically important for the absorption of vitamin B12, right? So whenever vitamin B12 reaches in the stomach, so it is present, it get bind with vitamin B12, and whenever they both reach in the terminal ileum, so hey, there it help in the absorption. Then also you need to write about the role of hypoxia, the role of liver and kidney, and also the role of very important hormone that is secreted by both liver and kidney, and how this erythropoietin is very, very useful for bone marrow stimulation, and how it can increase the RBC count also. So all these things are very important, and also the role of dietary factors, all the students should explain in the question. Okay. So this is all about the second one. Then the third one we have hemostasis. Okay, so in hemostasis, it can come, the question can come like this, that what is hemostasis, and also discuss it in detail. Okay, so now we will learn that what is hemostasis and why this is very important, right? So first, you need to answer that what is hemostasis? So this is the spontaneous arrest of bleeding by physiological phenomena. Okay, so what is hemostasis? This is the spontaneous arrest of bleeding by physiological phenomena. Okay, or we can also call the arrest or stoppage of bleeding, right? So basically, three stages are there. The first we have vasoconstriction. Then second we have platelet plug formation. Then we have coagulation of blood. So we will learn that how under these three stages the process of hemostasis will occur. Okay. So first we have whenever any blood vessel get injured, okay, so any blood vessel get injured, so there will be exposure of collagen, right? So exposure of collagen will occur, so platelets will try to form the adhesion, right? So at the site of injury there will be adhesion of platelets, right? And this adhesion will activate other platelets also to get accumulated at the site of injury, right? Now, with the help of this activation, what will happen? There will secretion of serotonin will start. So this secretion of serotonin will cause the vasoconstriction. So as the vasoconstriction, the process of vasoconstriction will start, so there will be reduced flow to blood will occur, right? As there will be reduced flow, so the loss of blood will be minimized, right? And along with this activation, the secretion of thromboxane A2 and ADP will start. So this thromboxane A2 and ADP are responsible for more aggregation of platelets, right? So as the platelet will aggregate, so there will be formation of primary hemostatic plug, or we also call it temporary hemostatic plug, right? Now, with the help of platelet phospholipids and with the help of tissue fluid, there will be formation of blood coagulation cascade, and finally, it leads to formation of thrombin and fibrinogen, and finally, it will lead to formation of permanent clot. So again, I'm repeating that whenever any injury occurs, the blood vessels constrict immediately and decrease the loss of blood and damage portion. Usually arterioles and small arteries constrict, and vasoconstriction is purely a local phenomena, right? So whenever any blood vessel is cut, the endothelium is damaged and the collagen is exposed, platelet adhere to this collagen and get activated, and the activated platelet secrete serotonin and other vasoconstricting substances also, which cause the vasoconstriction of blood vessels, and adherence of platelets to the collagen is accelerated by a very important factor, and that is known as Willebrand factor. Right now, next we have the platelet plug formation. So the platelet get adhered to collagen of ruptured blood vessels and secreted ADP. This ADP is adenosine diphosphate, and also there will be secretion of thromboxane A2. Now these two substances attract more and more platelets and also activate them, right? Now all these platelets aggregate together and form a temporary loose plug that is known as temporary hemostatic plug, which close the ruptured blood vessels and also prevent further blood loss, right? Now, finally, we have coagulation of blood. So during this process, the fibrinogen is converted into fibrin, right? As we can see, the fibrinogen is converted into fibrin, and fibrin thread get attached to the loose platelet plug, which block the further part of the blood vessels and prevent further loss completely, right? And this mechanism is known as stable hemostatic plug formation, right? So this is all about hemostasis. Okay, so if the question over the hemostasis will come, so you need to write down that what is hemostasis. You have to explain that what is hemostasis is the spontaneous arrest of bleeding by some physiological phenomena. Then you will write that various stages are there. First, we have vasoconstriction, platelet plug formation, then coagulation of blood. Then you need to draw this whole flow chart, which is the very, very important one. And also you need to each and every step, right, which has been written in this slide. Okay, now finally we have another question. We have covered function of blood, erythropoiesis, hemostasis we have covered. Now next we have fibrinolytic system. Okay, so talking about fibrinolytic system, so what is this system? So basically many factors that initiates clotting mechanism, right? Factors which are responsible for initiating clotting mechanism, they also stimulate the dissolution of the blood clot, and that phenomena is known as fibrinolysis, right? Now, fibrinolysis is the action of proteolytic enzyme, right? What are those proteolytic enzymes? Those enzymes are plasmin or fibrinolysis, right? Plasmin or fibrinolysis. Now, these are present in the circulation and converted to plasmin by the action of thrombin, okay? So, by the action of thrombin, they are converted, right? Now, by the action of thrombin and also important thing, that is TPA, right? What is TPA? TPA is tissue plasminogen activator, okay? And this TPA is released by tissue damage. Right? Now, plasmin lyses fibrin and fibrinogen with the production of fibrin degradation product, okay? That is known as FDP. And also, the process of fibrinogen is also slower than the clotting muck process. Okay, so this is all about the fibrinolytic system, and also this FDP is responsible for the inhibition of thrombin. Okay, so once again, I'm repeating all this process. So what is fibrinolytic system? So whatever the factors that are responsible for initiating clotting mechanism also stimulate the dissolution of the blood clot, and that is known as fibrinolysis. Now, fibrinolysis is due to the action of proteolytic enzyme, in which it includes plasmin, or the other name is fibrinolysin. Now, which get converted to plasmin by the action of thrombin and tissue plasminogen activator. Now, the TPA, which is released by the tissue damage that inhibit the formation of thrombin also, right? And this process is the slower, much slower than the clotting process. Okay, so this is all about the fibrinolytic system. Then, next we have the another question, that is lymph. What is lymph? Now we're going to understand what is lymph. Okay. So what is lymph? Lymph is an alkaline, clear fluid that is derived from interstitial fluid and flowing to bloodstream through the lymphatic vessels. Okay, so lymphatic vessels are those vessels which are responsible or which are the conducting pathway of lymph. So this is an alkaline fluid derived from interstitial. Now, we're going to learn that how the formation of lymph will occur and what is the origin of lymph. Okay, so as we know, lymph is formed by the interstitial fluid, right? So due to the permeability of lymph capillaries, when blood passes through the blood capillaries in the tissue, 9/10 of fluid passes into venous end of capillaries from the artery end, and the remaining 1/10 of the fluid passes into the lymph capillaries, right? Which have more permeability than blood capillaries. Okay, so whenever the lymph passes through lymph capillaries, the composition of lymph is more or less similar to the interstitial fluid, including protein content also. And protein present in the interstitial fluid cannot enter the blood capillary because their size is very larger. Okay, so these protein enter blood lymph vessels, which are the permeability to large particles also. Okay, so the cardiovascular system which pump the blood through its system, but it cannot return all the fluid from the body cell. Now what will happen? This lymph, the lymph pick 60% of the fluid dropped off at the cellular fluid. Now at this point, basically we're talking about the interstitial fluid. Okay, so now interstitial fluid pick up plasma and becomes tissue fluid also. And this fluid is then picked up by lymph capillaries. And finally, we get the lymph. So this fluid is basically lymph, that what we were talking about. Okay. So what do you understand by lymph? So after blood travels through the capillary bed and it's moved to venous system, some of its fluid is left behind the tissues, and this fluid is known as lymph. As we discussed, that lymph is a clear colorless fluid with a composition similar to blood plasma. It is nothing but the clear watery blood plasma leak out through the capillary wall to flow around the cells, and also it contains protein, glucose and blood cells. Right? Now, all the cells that bathe in the tissue, in the fluid, receive their nutrients and oxygen from it, and also tubular vessels transport lymph back to the blood, ultimately replacing the volume lost during the formation of the interstitial fluid. Now, lymph movement occurs despite low pressure due to peristalsis wall and compression during contraction of adjacent skeletal muscles and arterial pulsation. So whenever the question over the lymph will come, so first you have to answer that what is lymph. Then also you can state about various functions that also again we're going to discuss, that what is the function of blood. So first function of the blood is to return the protein from tissue space into blood. Right? Then next function we have that it is responsible for redistribution of fluid in the body. Then next thing we have that bacteria, toxin and other foreign bodies are removed from tissue through lymph. Then this lymph is also plays a very important role in immunity by transport of lymphocyte. Okay, so this is all about the function of lymph, and this question also can come in a short note. Okay, so the examiner can ask you, then write a short note on lymph. This is how the question can come. Okay, then we have covered all these 5 topics, and finally we reach towards the last part, the last question, that is function of platelet. So till now, a very important function of platelet we have already covered, and that was the clotted fraction, right? Sorry, the role, the hemostasis, right? So what is hemostasis? The hemostasis is the spontaneous arrest of bleeding, right? So now we're going to learn about the another major function of platelet. Okay, so first we have role in blood clotting. Okay, so as we know, platelets are responsible for the formation of intrinsic prothrombin activator. And here there will be beginning of the onset of blood clotting. And what is intrinsic prothrombin activator? That already we have discussed in the mechanism of blood clotting, right? Now next we have clotted fraction. So as we know, what is clotted fraction? In the blood clot, the blood cells, including platelets, are entrapped between the fibrin thread and cytoplasm of platelets contain a very important protein that is known as contractile protein, namely actin, myosin, and thrombostatin, which are responsible for the process of clot retraction. Okay, what is clot retraction? It is the reduction in clot size up to 40%. Right? Now, what is hemostasis? Already we have seen it is the spontaneous arrest of bleeding, and already the whole mechanism and the role of platelets we have discussed. Okay. Now, the next we have role in defense mechanism. So, the platelets are also very important for the defense mechanism also by the property of agglutination. By the property of agglutination, the platelets encircle the foreign bodies and also they destroy them. Okay. Then we have the role in repair of ruptured blood vessels. So the platelet-derived growth factor, that is known as PDGF. PDGF stands for platelet-derived growth factors, which form in the cytoplasm of platelets, is responsible for the repair of endothelium and other structures of the ruptured blood vessels also. So this is how the platelets has a very important role in the in the repair of ruptured blood vessels. Okay, so this is all about the major function of platelets. Now this is your turn to test yourself. Okay, so I know all the student knows what this diagram is showing the stages of erythropoiesis, right? So we're going to learn again that what are these stages. So first we have PUSE or hemocytoblast. Then next we have proerythroblast. Then next we have early normoblast. Then intermediate normoblast. Then finally we have late normoblast. And in late also we have two, the early one and the late one. And finally this stage is reticulocyte. And finally we have erythrocyte. So this is all about the stages of erythropoiesis, and already we know that as the stages proceed, the size of the cell will decrease, the size of the cytoplasm will increase, the size of the nucleus will decrease, and finally it get degenerate in the late normoblast. The appearance of hemoglobin occur in intermediate normoblast, and also the mitosis stops at late normoblast, right? Then state the major function of blood. So already we discussed that it help a very important role in breathing. Then we learn about electrolyte balance. Then pH regulation with the help of various proteins. Then we discuss about the role of nutritive function as it transport various hormones and enzymes. So another we write transport enzymes. Then defense mechanism with the help of WBC. Then also coagulation of blood. Okay, so this is all about the major function that is performed by blood. Then draw a flow chart of hemostasis. So already the whole process of hemostasis we have discussed, and also we know the various stages. So we have discussed whenever there is injury to blood vessel, what will happen? There will be exposure to collagen will occur, and there will be adherence of platelets. Right? Now what will happen? There will be activation of other platelets. And finally, there will be secretion of serotonin. And serotonin is responsible for vasoconstriction. So it will lead to reduce blood flow. And also activation of platelets will lead to secretion of ADP and thromboxane A2. And this thromboxane A2 is responsible for aggregation of platelet, and it lead to platelet plug formation. Right? Now, another thing we have learned that there is formation of prothrombin activator, and which leads to blood clotting or the formation of temporary hemostatic plug. Okay, so this is the stage 1, this is stage 2, this is stage 3. Okay, so this is all about the process of hemostasis. Okay, so I hope all the question to all the student are very much clear, and all the student now knows how to answer a question. So I hope you find this class helpful. So thank you so much.